So our last speaker, our last speaker in this session, as I mentioned before, is our visiting professor from Madrid, Rafael Correa. So this is an interesting story because when we were the CDTRP, launched in 2013, we had a series of projects that were taken on as our signature projects initially, and one of those was the study of regulatory T cells, and this was in collaboration with Megan Leving's group in Vancouver, the study of the possibility of using regulatory T cells from discarded human thymus in cellular therapy of with Tregs, as was going on in a number of places. So this is the continuation, this is the legacy of an international collaboration that developed based on that project. So Rafael has a PhD in medicine and surgery from the, well, I'm going to say this wrong, aren't I? Universidad Autónoma of Madrid. I'm so sorry. Lo siento mucho. And also a second PhD in biology from another Universidad in Madrid, receiving in both cases an extraordinary PhD award. His research career is focused on the field of immunopediatrics with a clinical translational approach. He's the funding chief of the Laboratory of Immune Regulation at the Gregorio Marañón Hospital Research Institute in Madrid. And the work of his group focuses on the study of immune homeostasis and the development of cell therapies to induce tolerance in the context of different diseases associated with immune hyperactivation. And so he's going to tell us about the steps that after we came to know each other, that took an original CDTRP project and has now moved it into actual clinical trials. So welcome, Rafael. We're very pleased you're here from warm Madrid to cold Alberta and BC.
Thank you very much, Lori, for your introduction. J'aimerais remercier les programmes de recherche de dons et transplantation du Canada. C'est pour mon plaisir de partager avec vous notre travail dans cet intéressant congrès. Thank you very much to the organizers. So, my team, as Lori introduced, is an international member of the CDTRP. And it's for me a pleasure to share with you the preliminary results of this clinical trial that, as Lori told, it was developed in collaboration with two members of the CDTRP. So I'm coming from Spain. It's true that we are not always good playing soccer or football, but year after year, we are the world champion in donation and transplantation. So as an example, Spain have the 0.6 world population, but we have the 20% of all the donations in Europe and 6% of donations worldwide. So organ transplantation is a big milestone of the medicine, but it's not a definitive solution because of the immune rejection. So in the moment that we transplant an organ, there starts a countdown and the immune system accomplishes their role and they recognize that there is a foreign element in the organism and they try to destroy with this immune response. So the current treatment is the use of immunosuppressive drugs. These drugs make possible the transplantation. Without this treatment, it could be possible. However, this treatment is not completely effective and moreover have a lot of side effects. And it's because the immunosuppressive drugs have a pleiotropic effect on all the immune system, not only in the response responsible of the rejection. And it's for that the chronic use of immunosuppressive drugs could increase the incidence of infections, autoimmune disease, or even cancer. So the result is that in some of the transplantation, the frequency of acute rejections in the first years could be higher than 20%. For most of the organs, we have a median graft survivals lower than 20 years. That is not a problem in the case that we transplant an adult individual of 40, 50 years old. These 20 years could be enough to reach the health expectancy of the healthy population. But as you can understand, in the pediatric context, there is a dramatic situation because the survival of this organ is very far for guarantee the life expectancy. So I'm coming from a hospital in Madrid that is the reference institution in Spain to perform pediatric cardiac transplantation. We have performed more than 200 pediatric heart transplantation. and probably we have responsible of more than 2% of all the pediatric hair transplantation that are performed around the world. So one of the problems that we find in this miracle of the hair transplantation is that half of the transplanted hairs in children are lost before 15 years after transplantation. So that requires a new transplant. It's very difficult to find organs in the pediatric context, so it's a big problem. And it's for that that it's necessary to develop new strategies that allow allograft survivals, inducing the immune tolerance and avoiding the toxicity associated to the immunosuppressive drugs. To achieve this goal, there are 3 golden rules for the tolerance induction, but unfortunately, we don't know any of them. So this is one of the goals of my research and the research of my group, to understand the mechanism of immune homeostasis and immune tolerance. We was investigating in different immune-related diseases, notably in the pediatric context, which are the immune mechanisms. And so one of the conclusions is that there is a balance between the 2 populations of the immune system, the effector cells responsible of the immune response, and a kind of cells that are regulatory T cells that have a suppressive role and have an important role maintaining the homeostasis of the immune system. So if there is a correct balance between these 2 populations, we can maintain the tolerance. But if this balance disappears because there is an increase in effector cells or because there is a decrease in regulatory T cells, is when this phenomena could appear, autoimmune disease, transplant rejection, or allergy. In fact, it has been proved that when a transplant and organ transplantation induce the effector cells that could be promote the rejection, in parallel, there is also the activation of regulatory T cells or specific regulatory T cells that could prevent this rejection. If this balance in the periphery between regulatory and effector cells is maintained, it's not a problem. So the question is why this natural mechanism of tolerance is not working in transplanted patients. And so this is the question that we try to answer. And we analyze in hair transplanted children that after transplantation, there is a progressive decrease in the levels of regulatory T cells. This is because regulatory T cells are very sensible to the immunosuppressive drugs. And interestingly, in parallel with this progressive decrease in regulatory T cells, we observe that there is an increase in effector cells. So we confirm that in hair transplanted children, this imbalance between Treg and TF could be one of the mechanisms responsible of the incidence of rejections in the first years after transplant. So clearly, interesting approach that could resolve this problem is a cell therapy employing regulatory T cells. If we increase the pool of regulatory T cells in the patients, maybe we can reestablish the balance between these 2 kind of cells. So this is an approach that has been tried by a lot of clinical trials in adults. All the clinical trials that perform in transplanted adults use the same approach. They obtain peripheral blood, half a liter of peripheral blood. They purify the regulatory T cells. They expand ex vivo, and after they reinfuse these regulatory T cells, autologous regulatory T cells, in the patients. So the results of the clinical trial demonstrate that this approach is safe, because it's the cell of the same patients, but we don't have still definitive results regarding the efficacy of this approach. One of the potential reasons of this low efficacy is because the quality of the regulatory T cells in terms of survival, suppressive capacity, and the stability of the phenotype is very dependent of the phenotype of the cells. So if we consider that in adults we have senescence or differentiated cells, so the quality is going to be of the starting material is going to be very low. So an interesting approach will be use AV regulatory T cells that are very frequent in children. However, the problem is that this approach is unfeasible in children because we cannot extract half a liter of blood in an infant, logically. So at this point, we decided to explore new sources to obtain regulatory T cells, maybe placenta, maybe cord blood, maybe a thymus. And it was at this point that I met Lori West in Madrid. And so she told me about a manuscript that they had been submitted that describe that effectively the thymus could be an excellent source to obtain therapeutic regulatory T cells. So The thymus is an organ that evolves with the age, is very functional in the infancy. And so I have to communicate you that all the people that are in this room, our thymus don't work very well. So in children, it's very functional, but moreover, the thymus covers completely the heart, and it's for that in infants with congenital cardiopathies, the surgeons must remove the thymus to have access to the heart. And because this thymus is not possible to reimplant in the patients, normally this thymus is discarded. Is thrown to the trash. So for us, it was an opportunity to explore if this tissue that is discarded in the theater, we could use it to obtain regulatory T cells. So for that, this was the project of Esther in my team. We adapt the original protocol to our facilities and doing some modifications. And we were surprised because with this protocol, we obtained finally a highly pure product of regulatory T cells. The purity was higher than 95%, and the viability was also very high. But the biggest surprise was with the yield. So normally in 20 ml of peripheral blood in a child, maybe we can obtain a maximum of 2 million of regulatory T cells. In the adult peripheral blood in half a liter, probably the maximum is 30 million. But from a single thymus in some of the patients, we obtain 13,000 millions of regulatory T cells per single thymus. So that was amazing. It was an amazing quantity. So because this approach, we resolve some of the limitations of the original approach. So in comparison with the standard approach in the peripheral blood, we obtain much more regulatory T cells. So we can produce more therapeutic doses with this approach, but moreover, because these cells are not differentiated, are just being produced in the thymus, the features of these cells are much better than the adult cells in the peripheral blood in terms of survival, suppressive capacity, and moreover, this approach is feasible in children. And another interesting point is that we have seen that these regulatory T cells of the thymus are not immunogenic, and so it could be used as an allogenic and off-the-cell use of this therapy. So at the light of the potential of this therapy, we decided to move forward from the bench to the patient. We started a clinical trial. So one of the big challenges when you want to develop a clinical trial, an independent clinical trial that do not use a drug that is commercialized by the pharmaceutical company, is to obtain the funds. So it was quite hard to knock all the doors in Spain to the government, to the private foundations to obtain the funds. But finally, we had success. We obtained around 5 million euro for public funds. And so finally, we developed this clinical trial. It was also very difficult to convince to the Spanish medical agency about the safety of this approach because it was the first time that it's going to be used. So for us, it was more than 1 year discussing with the medical agency. And finally, we obtained at the end of 2019 the authorization of the Spanish medical agency, and we start recruiting patients in 2020. So this clinical trial is pioneering because it's the first one performing a third therapy in transplanted children, but notably is the first in humans that have used these regulatory T cells that are obtaining from the thymic tissue. So in our trial, we have recruited 10 patients yet. We have treated 4 patients with this thyTreg, and the goal of our trial is to treat at least 10 patients. So the protocol is very simple. We have candidates, and after the informed consent from the parents, they are included in the clinical trial. When this baby receives a heart transplantation, the surgeons must remove the thymus, and we take this thymus in our GMP facility to produce the therapeutic dose of thyTreg. We wait more or less 1 week to leave that the patient become stabilized in the ICU. And after 1 week, we infuse intravenously the regulatory T cells of the patient that in this case are autologous because they are coming from the thymus. And so these are the inclusion and exclusion criteria. In the exclusion criteria, we cannot include the George syndrome patients because the thymic function size is modified. We don't include HIV positive patients. And in the inclusion criteria, we have selected infants younger than 2 years old because we have seen that in this patient, the thymus provide an excellent product and an excellent yield and regulatory T cells. So these are the 4 patients that we have treated until now. So this we call the miracle patients because they have received this therapy, but some of these patients also have received an ABO-incompatible transplantation that, as you know, Lori have a lot to say about that. So these are the 4 patients that we have treated. And I would like to be pleased to present the preliminary results of the first patient. So regarding the product that we have infused in the patient, we obtained a very high purity, but the features and the phenotype of the product that we have infused are excellent. Here you can see that our product produces high levels of interleukin-10, that is suppressive cytokine, but they don't produce pro-inflammatory cytokines. The suppressive capacity of the cells is very high, more than they inhibit the proliferation of CD4 and CD8 cells that are the principal mediator of rejection in more than 75%. And moreover, we also analyze the stability of these cells because one of the limitations in adults that regulatory T cells, when they are in a context of pro-inflammatory environment, the pro-inflammatory cytokines can switch the phenotype of the regulatory T cells, and they lose the suppressive capacity and acquire pro-inflammatory role. So we tested if our thyTreg also have this instability of the phenotype, but we was very surprised because when we culture these cells in pro-inflammatory conditions, the FOXP3 expression is maintained completely intact. Also, the suppressive capacity is preserved. And more interestingly, these cells, even in pro-inflammatory conditions, never produce interferon gamma or IL-17. So the stability of the phenotype is very high. And finally, this is the results of the third patient that we have treated. The first patient, it was a girl of 6 months old. And so in blue, there is the evolution of our control group. Our control group are infants, also younger than 2 years old, that have received a heart transplantation, but do not receive the thyTreg therapy. And as we described previously, in this patient, there is a progressive decrease after 1 year and 1 year after transplantation in the regulatory T cells levels. So that could provide this imbalance between the 2 cells. But as you see in our first patient, we increased the reserve for regulatory T cells, but interestingly, this reserve for regulatory T cells is preserved 1 year after the single infusion that we perform of cells. So Cici Irene is the first girl in the world that has received a thyTreg therapy. These photos are 1 year after the transplantation and 1 year after receiving the thyTreg therapy. And so the results to date is that we didn't observe any complications attributed to the Treg administration. There are no suspicion of graft rejection episodes in these patients that are relatively frequent in the first years after transplantation. And interestingly, we didn't observe nosocomial or opportunistic infections that are quite frequent in the period that they stay in the hospital, maybe because we are trying to recover the correct immune homeostasis with this therapy. And so the feasibility and the safety is yet confirmed because in the 4 patients that we have treated, there are not any adverse effects attributed to the therapy. And we have preliminary results, but we have some evidence that the repopulation of Treg cells with this therapy could be possible. So, which is our challenge? Our challenge is a transplant for life. So the next step in this project is that we would like to test if we can reduce the immunosuppression in patients that have received this thyTreg, because we have seen that we can maintain without rejection episode the patients. Another approach that we would like to explore is to use allogenically these cells to treat adults, transplanted adults. And why not? That could be an interesting approach that could be useful in the xenotransplants context. So moreover, we obtain so many doses from a single thymus that we have cryopreserved all the regulatory T cells in these patients. And maybe in the future, we can do new doses of their own regulatory T cells to maintain the survival of the graft. But moreover, we think that these cells constitute a big arsenal that could have infinite possibilities treating immune-related disease. And it's for that that we are exploring not only in transplantation, but also in other diseases, including COVID, because as you know, the main cause of death in COVID patients is not the virus, is the hyperimmune activations related with the response against the virus. And so it's for that Marta, that is a postdoc in my team, is exploring the capacity if the thyTreg could be employed as an off-the-cell treatment in the allogenic context. So first, she confirmed that the suppressive capacity in the allogenic context is preserved in the thyTreg. They also inhibit CD4 and CD8 cells coming from a different individual. But more interestingly, when we analyze the immunogenicity capacity of these cells, because this is the big problem of the allogenic use of cell therapies. If you put this setup and the cells from an individual in a different subject, these cells are going to be recognized as foreign cells and are going to be destroyed, making the therapy unuseful. So it's interesting that maybe because these cells are very mature, we observe that they don't induce an allogenic response in the receptor. So we perform co-cultures, and so in green, we can see our control conditions. We put cells of the same individual, and there are not proliferation, there is a basal activation, and there are not interferon production. If we mix allogenic cells from different individuals in blue, we observe that there is a strong immunogenic response, increasing the proliferation, attenuation, and interferon production. But if we co-culture this thyTreg with PBNCs from another individual, there are not any proliferation, activation, or interferon gain production, even if the cells are irradiated. So that demonstrates that these cells are invisible or have a very low immunogenicity in the allogenic context, even if in our co-cultures we have a big mismatching regarding HLA. So it's for that now we are trying to develop a new clinical trial. We have yet submitted all the documentation to the Spanish Medical Agency, and our idea is to take all the cells that we can obtain in the pediatric thymus and use this therapy for adult COVID patients that are in the ICU, and to test if this therapy could be safe, of course, and also to test if it's effective reducing the cytokine release syndrome or reducing the time that the patients are in the ICU. So we expect that in the first months, we will start this interesting clinical trial that will be the first use of regulatory T cells allogenically, because in the cord blood has been yet used with success in clinical trial, but with certain HLA compatibility. And so this is the project that we would like to develop now. So I would like to finish. Thank you to all my team, also all the departments in the hospital that have participated in the project, and of course, the collaboration of Megan Levins and Lori West in this project. Thank you very much.
So, nice overview, very, very nicely done. I realize that I have neglected to acknowledge the work in the initial project of Dr. Esme Dyke, who really led the initial project and continues to lead complementary collaborative work, also looking at the immunogenicity of regulatory T cells, but from a different, of the thymic regulatory T cells, but from a slightly different perspective. So these continue to be complementary studies that go on in different places around the world. I was just going to say something else. Maybe I'll come back to it. Mel. Mamata.
Yeah, that was an excellent talk. Thank you for sharing this experience with us. So can you comment on the impact of Tregs on fibrosis progression? Like, say, I'm guessing that cardiac graft fibrosis has an accelerated rate of progression, like in the liver, it certainly has an accelerated rate of progression compared to the native liver.
Yeah.
So I'm guessing it's the same in the heart. Like if you have ongoing immune activity, there's a certain rate of fibrosis progression that is probably accelerated, and hence the reduced duration or the reduced graft lifespan. So what are your thoughts on the impact of Treg therapy on graft fibrosis?
Yeah, it's a very interesting question. I'm not an expert, but I know that fibrosis, there are some evidence that point out the possibility that could be an autoimmune response, or there is the hyperinflammation associated to the radiation could also promote the fibrosis. I think that if you have a rejection and a good rejection in the first years, then the risk of having fibrosis after is increased. So maybe for this reason. And of course, the therapy with regulatory T cells could be very interesting also to prevent that. So we don't know this still because in this patient we don't have enough follow-up. But we have seen that in comparison to adults, in adults, the therapy with regulatory T cells has been observed that you have an increase in the Treg values in periphery, but after 1 month, this increase disappeared. So you have seen this result that maybe because the high survival of these cells, we maintain this increase in regulatory T cells, and so the protective role in fibrosis on all immune-mediated response could be prolonged on time, and maybe could have also a preventive role for that. Yeah.
It's like a good novel, this story. You can't wait for the next page. And I think you gave an excellent next page. I'm amazed by the stability of even in inflammatory condition of these cells. You know what the trick is? Because there's several reports, showing plasticity of expansion culture. So what's in your expansion culture that makes it so stable and for them to commit to FOXP3 and their Treg phenotype?
Yes, in that experiment, we use the normal conditions that are published to induce a switch in the phenotype of the cell. So we use IL-17, IL-2, and so a cocktail of different cytokines that are described to induce the switch. And so even in this cocktail, in this culture, so we have seen that the features of the regulatory T cells are preserved. So these are in vitro results. We cannot be sure that in the in vivo context is going to happen the same. But at least in this culture and in this analysis, we observe that it's stable. And we have also confirmed that the methylation of the FOXP3 is also preserved. So normally the problem with the differentiate regulatory T cells if that in this condition there is a methylation of FOXP3, and it's probably the reason that they lose the suppressive phenotype. But in our hands, with these experiments, even the demethylation of FOXP3 is preserved even in that conditions. Yeah.
Excellent. Maybe another one quickly. When we want to use off-the-shelf therapy, you need to cryopreserve. And I know there was some work on cryopreservation. Here you transplant fresh, right? Right. So have you tested if you cryopreserve them, are they as potent?
Yes, Lorian also is working in that cryopreservation. So we have very interesting results because we cryopreserve the GMP cells, and we have taught the cells after 8 months cryopreserved in liquid nitrogen. And interestingly, all the features are maintained. So we recover a population with a very high viability, higher than 90%. The suppressive capacity is preserved, higher than 75%, and also the phenotype. So we have now to evaluate at long term the caducity of our products. So now we have aliquots that have cryopreserved, and we will show after 1 year, after 2 years. So I think that, as you told, in this first clinical trial, we are using fresh cells because we don't need to cryopreserve. The timing is perfect. But for the off-the-cell use, clearly we have to cryopreserve the cells. And I think that is possible because in our experience, they maintain intact their properties. Yeah.
Excellent. Thank you.
Maybe I could just for a moment pick up on, is this on?
Yeah.
Just to follow up on those two questions and comments before we come to Tom and Elaine. So the work that ESME is carrying on with cryopreservation specifically looks at ice recrystallization inhibitors, which may really, even though so far DMSO seems to work adequately, but we may have even superior results using selective ice recrystallization inhibitors. And that work is ongoing. And again, that's led by ESME. The inflammatory conditions. So the work that ESME did originally also looked at whether these cells are stable under inflammatory conditions in terms of their phenotype and their function. And so your work nicely kind of confirms that again, because as you said, these are new cells going into new trials and so on. So it is helpful that they don't take on a phenotype that could be destructive. And finally, the work that Megan and Esme and our group are doing in Vancouver and in Alberta as part of the greater CDTRP envelope of this project. As you say, it's like a novel that continues, really is also aiming for a clinical trial in GVHD. And that's using the cell manufacturing facility at the University of Alberta, looking at these. So it's a nice complementary set of experiments. Sorry, Tom.
Oh, that's great. Tom, that's from BC Children's Hospital. A couple of questions. One's just on the safety side. What was the donor and recipient mismatch as related to viruses like CMV and EBV? Maybe I missed that. Just the concern being that if there's a donor-derived infection with a herpes virus at the time of transplant, and then you're giving them Tregs, you may develop tolerance to those viruses as well.
Yes, an excellent question. So the answer, I think that is difficult because we don't have still the experience, but the hypothesis is that regulatory T cells only provide the suppressive role in the place that they detect an hyperactivation of the immune system, and only in the scale that they detect this hyperactivation. So in a healthy donor, in a healthy individual, regulatory T cells are there, and I don't inhibit the defensive response against the virus. So in our clinical trial, we never overpass the maximum quantity of regulatory T cells that are in periphery. So we only restore the regulatory T cells that the transplanted patients have lost by immunosuppressive drugs. So we don't expect that we have a over immunosuppression of the immune response against the virus for this treatment, because this natural mechanism of these cells to maintain the immune homeostasis, and because we don't never overpass the quantity of regulatory T cells that are normally in periphery.
So I mean, I guess they were infant donors as well as recipients, so they probably were EBV and CMV negative anyways.
Yeah.
And then sorry, I had to step out to take a call, but I just didn't hear the immunosuppression protocol that you're using, in particular thinking about the calcineurin inhibitors and whether you had any concerns about CNI exposure on the Tregs.
Yeah, the protocol that we use in this clinical trial is the current protocol in our institution, but we have removed the induction therapy because we published a work that demonstrates that basilizumab have a very strong effect in regulatory T cells. Yeah. And so there was not so many evidence that is required and is imprescindible this induction therapy. We was speaking with Lori, and for example, she told us that in Canada, not all the hospitals are using this induction therapy in her transplanted children. So at the level of the result, we decided to remove the induction therapy. And after, both the control group and the treated group received the same immunosuppressive regime, because we don't have still evidence that we can remove or reduce the doses of the immunosuppressive drug. So we have removed the induction therapy, but now the next step is maybe in the patients that have already received the therapy, trying to reduce the immunosuppressive regime.
Thank you so much.
Thank you.
Great.
Thank you.